the last 150 years or so. And that is just incredible if you think about it, how it would change one's whole attitude toward life. It gives a different sense of the permanence of life and the likelihood that you're going to be able to live long enough to achieve something. That's what science has done for us, and nobody should ever forget that. Still, the situation is today that about 1/4 of all people born on the planet will ultimately die of an infection. And that's what the Microbe Hunters is about, and that's what my work is about. And that's a lead-in to what we do every day. We're ultimately trying to solve this problem that so many people die of infectious diseases. Infection isn't like most other causes of death either, because you see it tends to kill people at a very young age. The red bars here indicate infectious disease, and the green bars indicate all other causes of death. And in old age, other causes of death certainly predominate, but if we're looking in the developing world, and we look at young people, 0 to 4 years of age, even up through the age of reproduction, which I take to be about 44 or 45, then infection is the dominant cause of death. This is the thing that claims more lives, and certainly more years of human life than anything else. You've all had infections. You've had colds. You may have skinned your knee. You may have had an infected thumb. You may have had appendicitis. I don't know. But infections all have things that are a bit in common, and here are a bunch of them. Some of them are really pretty trivial, like this one, which is called tinea faciae. It's just sort of a rash. It will go away. Others can be very, very serious. This is Neisseria Meningitidis, sepsis with a gram-negative bacterium. That's often fatal, and it too happens a lot in young people, older sometimes. And this is a staph infection, herpes infection, chlamydia infection of the eye, and measles. They all look different, but they've always been known for thousands of years because people spread them from one person to another, and because they're painful often, and they have an inflammatory character. But today we all know, of course, that they're caused by germs. All of those things are caused by germs. And that wasn't always known. It's just incredible to think about. But in the 17th, midway, even through the 19th century, midway through the 19th century, people didn't know what caused infection. They thought it might be caused by being in a bad air, or by being in the wrong place, or by being in dirty environment, but no one connected them with germs. This man was named Antony von Leeuwenhoek, and in the 17th century, he had already seen germs with a microscope that he made with his own hands. He was a lens grinder, and he looked at scum from his teeth, and he saw bacteria that looked like this, and he drew pictures of them. He published his work, and nonetheless, he didn't realize, it didn't occur to him, that this was a cause of infections that doctors had always been seeing and coping with. It wasn't until the mid-1900s that these two men, Louis Pasteur and Robert Koch, jointly formulated the germ theory of disease, as we call it now. And we now know that everything that we recognize as an infection, by and large, is caused by a bacterium, a fungus, a virus, or a protozoan. Different forms of life, but very small forms of life. It began to be a question then, what is it that's recognizable about an infection? How does our immune system recognize that we have an infection and begin to mount a response to it? And by the early 20th century, certainly, the concept of receptors was well entrenched in biology, and people began to think there must be receptors that recognize microbes and tell us when we have an infection. There was a man named Richard Pfeiffer who worked with Robert Koch, and Pfeiffer coined the term endotoxin to describe something associated with cholera germs that was very toxic. And these germs he established as an infection in guinea pigs. He could boil them, and still injected the dead bacteria, the guinea pigs would develop a fever and they'd go into shock and they would die. So something was intrinsically poisonous about the bacteria. So we might say the progress, intellectual progress, by the year 1900 was that first of all it was recognized that infectious diseases are caused by microbes or germs. Germs make substances that harm us, such as endotoxin, and the big question at the dawn of the last century was, what is endotoxin and how does it harm us? Well, I won't tell you the whole story because it's really very involved, but it was recognized that endotoxin was a molecule called lipopolysaccharide, and eventually this was found to be a major component of gram-negative bacteria, which means about half the bacteria that exist on the planet. And the structure of endotoxins, the chemical structure, was eventually solved. And it was found that they had a part of that molecule. Endotoxin was made of a lipid A moiety and a bunch of polysaccharide, or sugar chains. And it was the lipid part that was really toxic, that was doing all of what fiber saw. And we might draw it today, LPS, as this molecule. And for the next, let's say, 80 or 90 years after Richard Pfeiffer did his work, nobody knew how this LPS acted to cause toxicity. What was known was that it could act on macrophages, which come from white blood cells in our body. And the LPS would do something to the macrophage to activate them. And it was one of my own early discoveries that these cells would make a protein called TNF. And TNF was a mediator of endotoxic shock. It would bind to receptors all through the body, and it would cause severe inflammation. And that was how endotoxin was poisonous. But the big question was, what's the receptor for LPS? How does that actually trigger a response? Because that's where everything began. And that problem was solved again in a really complicated way. We worked on it in my laboratory for about 5 years, and our work was based on these mice that you see here. It turned out that there were animals out there, among many strains of mice, that wouldn't respond to LPS. They didn't recognize it. You could give them any dose you wanted, and they wouldn't get sick, they wouldn't die, didn't develop fever. And using a pure genetic method, we tracked down a mutation in these mice that made them different from all other mice. And that mutation was in the molecule you see here. I'm going to make it spin around for you. That molecule is called toll-like receptor 4. You see the blue molecule is TLR4. There's a smaller molecule bound to it in purple here called MD2. And together these act as the receptor for LPS, which fits into a basket in the MD2 protein. And this causes a little tiny change in shape of this molecule, and that triggers a signal inside macrophages and makes them activate the TNF gene and hundreds of other genes as well that are what cause inflammation. It was later learned that not only TLR4 exists in our bodies, but there are many TLRs, actually 10 of them in all humans. And each one of them recognizes a different molecule that comes from microbes. This one I just showed you, TLR4 recognizes LPS, but TLR5 recognizes a bacterial protein called flagellin. TLR3 detects double-stranded RNA that's made by some viruses. TLR2, together with TLR1 or TLR6, will recognize lipopeptides made by bacteria or fungi, and so on. So collectively, these receptors inform you when you have an infection. And they don't only cause shock, but they cause good things to happen as well. Without them, you wouldn't ever develop a good immune response, and you would die of any infection you got. You can look at proteins like the toll-like receptors, and you can organize them by their similarity to each other. And we soon saw that all the mammalian toll-like receptors were quite Drosophila, there were also toll-like receptors. And even in plants, there were molecules like that. And it emerged that if you alter one member of that family of molecules in the fruit fly, or in mice, or in humans, or in plants, you get a situation where the animal or plant can be overgrown by an infection. So here you see a fruit fly that's been infected with a fungus. It can't reject the fungus because it's missing toll, the namesake of the family. In humans, a mutation in TLR4 will make susceptibility to a gram-negative infection. In the plant, like in the flax plant shown here, the plant will be overgrown by a fungal rust that you see growing on it. So clearly this is a major way of recognizing microbes. You can think of these receptors as a kind of barcode reader that tells what sort of microbe is infecting your body, and they mount the correct response to deal with the infection. By the year 2000, all that work was done, and I could say that these were the questions before. The new understandings were that endotoxin is lipopolysaccharide, or LPS. LPS activates the release of toxic cytokines like TNF. The receptor for LPS is TLR4, and other TLRs detect other molecules made by microbes. And finally, the new question might have been at that stage, what other essential immune defense proteins are there? And how do the toll-like receptors signal into the cell? These were things we wanted to understand. And we took a very special approach to try to solve those questions. Does anybody know the word serendipity here? Nobody? Yes, use it in a sentence for me. Pardon?
Doesn't it mean like spontaneity?
Spontaneity? Yeah, kind of, just right. It means a kind of random discovery. That's serendipity.